Hi, my name is Lauren. I work here as a researcher on the GBNERR team, and I have the pleasure to introduce our next block, which is all about eelgrass. We are going to have two presentations in this block. First up will be Trevor Mattera. He is a habitat program manager with PrEP. And then he will be followed by Dr. Theresa Oehmke, who is an assistant professor of mechanical engineering at UNH. So Trevor, if you would like to start first here.
Great, thanks, Lauren. Let's see. We'll back it up. All right, good afternoon, all. Like Lauren mentioned, my name is Trevor Mattera. I'm the Habitat Program Manager at the Piscataqua Region Estuaries Partnership, or PrEP. And I get to kick off this short and sweet eelgrass block with you, the best habitat. This is really just going to be sort of a high-level overview of some of the goings on in eelgrass restoration here in Great Bay. Because I get to kick off the talk, I also get to talk about some of these cool questions like why we love eelgrass in the first place. As some of you may know, and the rest should, eelgrass itself is a flowering angiosperm that's actually totally subtidal. It likes to be anyway. Sometimes it's intertidal, but for the most part, it likes to be completely under the water. It is not a seaweed. It is actually a photosynthesizing plant. And because of that, things like sunlight is actually very important to it, but it provides a lot of habitat benefits for us. For instance, housing all of these super cute little critters here. It acts as a nursery for a lot of different fish and crustaceans, as well as invertebrates. And it does a lot of other things for us as well. For instance, stabilizing shoreline, dampening wave energy, just really, really great stuff all around. It makes the basis of our subtidal ecosystem here in the estuary and our near coastal environments. And because of that, we at PrEP do a lot of work monitoring the health of this habitat and this resource. We have multiple different monitoring protocols, ranging everywhere from aerial monitoring using both low-flying aircraft and drones, as you can see here, to in-water monitoring, where we get down and dirty, down in the eelgrass beds, measuring different health metrics to see how these beds are doing. A map of 25 of our sites is actually down in the bottom right here. You can see some of the places where we're actually in the water and keeping track of how this is doing. Which then begs the question, well, Trevor, how is the eelgrass doing? And as many of the smart people in this room, which is everyone is aware, if it was doing great, I wouldn't be up here in a habitat restoration work group with you. So, as you can see, we are seeing a disturbing trend in terms of eelgrass over the years. Back in the mid-90s, we had somewhere around 3,000 acres total throughout the estuary. That's both Great Bay proper, as well as our coastal habitats out in Portsmouth Harbor and Little Harbor and Little Bay. And you can see that's been sliding really through the early 2000s. In 2006, I'm going to use the pointer again, wish me luck. 2006 here, you can see that Mother's Day storm really took a whack on our total acreage. But then it sort of stayed pretty stable with little ups and downs throughout the like the 2010s, 2010s, until this big one here in 2023, where we saw a very significant loss, one of the biggest losses we'd seen in decades. And again, that, if you remember, you couldn't really go outside that summer at all because we were having just record-breaking rains throughout July, and that really did a number on the eelgrass's growing period. 2024, we didn't really see much of a recovery from then. And although I don't have data for you, finalized data anyway for 2025, anecdotally, I can tell you that 2025 was probably a really bad year. A lot of the beds that were even around in the end of 2023 didn't really come back in 2025. So I'm a little worried to see what that final number would look like. Anyway, just because sometimes looking at dots isn't as good as looking at a map, I wanted to actually show you the visualization of what 2023 looked like. So just to get your eyes trained on it, the green is the existing beds in 2023, and the yellows that's surrounding it were beds that were there in 2022. So this represents hundreds of acres lost in Great Bay alone. And again, in 1 year. So not good. But I do want you to sort of hold this in your mind as I do the rest of the talking, because it really paints, it puts what I'm going to be telling you into a different perspective. So why I'm up here? Can we restore eelgrass? There are many common restoration techniques when it comes to eelgrass. People have been using them for decades. I will tell you that in most cases, what it amounts to is taking adult shoots from healthy beds and picking them up, and just like you do in your garden, going to where you'd like it to grow and just sort of planting it and then stepping back and praying. A lot of the different methodologies we actually talk about and see are really just ways of stabilizing those adult transplants down to the sediment that you want them. So up in the top left, we have something called a turf, which is transplanting eelgrass remotely with frames. Down here is what we call a tortilla method, where you string it through burlap. And over here, we have something called horizontal rhizome technique, where you take a couple of eelgrasses together, you say, you two are going to live together, and you staple them to the sediment. Right in the middle here is something else where I mentioned it's a flowering plant. This plant also produces seeds. So seed-based restoration is also a technique that is possible to use. We've been trying to do this now for, well, for a number of years, but I've been trying to do this now since about 2021, where we started doing some pilots. Here are a bunch of pictures of some of our amazing volunteers doing some of the pilot work. And then in 2022, going into 2023, the Municipal Alliance for Adaptive Management, which is a collaboration of communities here around the Bay, secured some congressionally designated funding for a large-scale oyster and eelgrass integrated restoration project. I worked with Dr. David Burdick and Dr. Ray Grizzle on this, as well as many of the folks in his lab. And we worked over 2023 through 2025 on this project. You can see some of what we were doing here, including the 3 sites that we worked on, from the Lamprey River in 23 and Nanny Island in 23, Nanny Island in 24, and Herod's Cove over in 25. And we used some different techniques. In 2023, we were using completely horizontal rhizome techniques. So again, harvesting a lot of plants and getting in the water and stapling them down individually in these 2 sites. You may now go back to that mental map of 2023, where we lost hundreds of acres in this area. And I can tell you, unfortunately, that the 1/5 of the acre that we put down wasn't spared either. In 2024, we know we needed to pivot for a couple of reasons. First of all, because of what was happening in 2023, and also because trying to scale horizontal rhizome is actually exceedingly difficult. It's, again, you're planting these down individually. It's a lot of in water time. And because of that, we use something I'll be calling a modified turf, which is essentially a turf and tortilla hybrid. There it is, which we sort of came up with on the fly, because the turf itself is a weighted frame, which keeps that grass down. It offers a little bit of stability to your plantings, where the burlap itself will actually get under and biodegrade after a certain amount of time. Through the pilots I talked about in 2021 and 2022, we saw that even though the turfs were doing a great job of holding those plants down and offering some stability as they were establishing themselves, you had to go back and take them out. And generally, the bottom of these things are the wire that you tie the plants to, and you would often pull up 1/3 of the plants that were, happily down in the dirt. So this we found was a happy medium. And we did this for both 2024 and 2025, and I'm going to show you some results on those. So in 2024, we went out 3 weeks after. Again, this was the first monitoring period we had on those plants, again, because you had to remove the frames after you put them down. So 2 to 3 weeks is generally when you put them down. And we were seeing around a 25%, with a good amount of variability in there, 25% survival across planting units, generally the turfs. And toward the end of the growing season in 24, although your median is again down 0, you do see some success in there, going up towards about 40-ish% there. So there was some grass still growing in 24. Great news. 25, we're like, okay, first of all, we weren't super impressed with the survival of Nanny Island, so we wanted to try a site that was closer to one of the beds that was hanging on through 23. In Herod's Cove, it had a little bit more protection from some of the wave energy there. And you can see from our 3-week monitoring that suddenly we're up in the 75% range when it comes to survival. Unfortunately, again, like I mentioned, 25 was not a good year for eelgrass, and because of that, after that sorry, got distracted with the one. We didn't see any kind of survival after that when we went after a number of months. So one of the things, again, we tried pivoting towards is seeds. In 2024, we started this as well with a phenology, and the big question is, when are the seeds popping up? Eelgrass seeds come up on a pretty well-defined phenological cycle where you're looking for a stage 4, they're called spathe here, where your developed seeds are going. So we had phenology surveys across 5 different sites in the estuary. And you can see here, these coastal sites up on top, your stage 4s are really sort of peaking around early August. It was really sort of spaced out when it comes to the Great Bay seeds, and you even see peaks as far out as October, but the green and the blue here is the number of developed seeds, and you can see your seed counts across shoots is just much higher than what you're seeing in Great Bay. So we did harvest from the coast in 2024. We put them out in November 24 and April 25. And just to show you some preliminary numbers of that, although we were seeing, again, very bad eelgrass conditions in 2025, we were seeing a little bit of higher numbers across our seeding areas, walking 10-meter transects in each of these as compared to the unplanted control. So with that, next steps for us is really more seeds. We're really trying to look into this as a restoration technique. We have seeds from the 2025 harvest going out on Monday to be dispersed in the same area. We'll be looking at germination rate studies. We are also awaiting a decision on a proposal for funding this work into the future, into 2026 and beyond. And we are also working with a lodge of regional partners, because I can tell you that states all the way up and down the East Coast, as well as over on the West Coast, are really looking at seeding as a viable restoration technique. And we're all struggling now with the same questions as to how we do this and how we can bring this resource back. With that, thank you so much. Contact me for any of your eelgrass needs. Huge thanks to MAM and a lot of the people I work with, including David Burdick, Cynthia Hayes, and all of our great volunteers, and thank you very much.
All right, Trevor left us with a great question of how to use seeds to restore eelgrass, and Theresa here will hopefully tell us a little bit more about that.
Hi, thank you for having me. This is maybe the most interdisciplinary audience I've ever presented to, so please ask me questions if I skip over anything. Trevor did leave us with a very interesting question, and I will show what work we've done so far to try to answer some of those questions. So today I'm going to be talking about some of the, I stepped on something. Today I'm going to be talking about some of the experiments we did to look at how these eelgrass seeds move through different types of vegetated canopies. The work that I'm going to be showing was all the data was taken by Jade Hodges. The analysis was done by Will, who is over here, and it's part of funding with Cynthia, who's here, Tracy, who's here, and Tom Lipman. Great, so thank you, Trevor, for the great introduction to eelgrass and Great Bay Estuary. We went out and sampled in the estuary in 2023, I think. And so these are some of the pictures from the sampling time, and a lot of the experiments that we did were based on the data that we collected that summer. Great, so to motivate kind of the talk on whether or not even looking at the seeds is good, I'm going to show some data that Cynthia collected from these 2 sites. So one is at Portsmouth Harbor, and the other one is at this site in Great Bay Estuary. And just very quickly, since we have already had some background on the estuary, what we see is that inside of the estuary, we have here more genetic diversity in the eelgrass stems that were sampled compared to the ones that were in Portsmouth Harbor. So that means that we expect to have kind of these eelgrass in the estuary that are growing from seeds. So kind of the first question that I was even curious about before starting the experiments was, is this actually viable? If the eelgrass are just reproducing asexually in the estuary, why are we trying to use seeds? But we see with the genetic diversity that there is eelgrass growth from seeds. It's like, great, now I can do my experiments without having to kind of come up with a different motivation. So when we were sampling over the summer, we took a bunch of data. So I'm going to show you kind of how we took that field data into the lab, but I want to focus on the hydrodynamics. So we'll show that last, but we have all of these different meadow geometry data. We have these numbers that tell us about the plant geometry. So that plant geometry is basically telling us how tall the plant is compared to the water depth and how wide those plant blades are compared to other things like the plant height. So what that ratio is. And then our meadow geometry tells us how closely those plants are spaced together in different types of eelgrass meadows. So if it's really close together, then we'll say it's a dense meadow, farther apart, a sparse meadow. And finally, we have all of these flow fields. So these are the ones that these last ones are the ones that we focused on for our experiments, because these are the ones that are easiest for us to change in the experimental facility that we have. So this is how fast the flow is moving. It is also how big the eelgrass seeds are compared to kind of the flow forcing, and also how the eelgrass seeds move or turn over or rotate compared to the flow forcing as well. So that's really what we're looking at. Our question is, how do we look at the eelgrass seeds and the flows that they're in? So that's that meadow vegetative flow, and what happens kind of over time with different flow speeds to how those seeds move. Okay, so this is the facility we used. It's a 10-meter long water tunnel with a cross section of 40 centimeters wide and 30 centimeters deep. So I'm there with Tracy for scale, in case anybody had not yet seen it in the scale size. It is taller than both of us. And we made these mimics of our eelgrass, and this is what we tried to kind of replicate was a sparse meadow. So when these eelgrasses or these eelgrass clusters are really far apart from each other, mostly because it takes a very, very long time to cut all of the mimics. And so we cut enough for sparse meadows and are still waiting or working on cutting more for the more dense ones. We also have these kind of mimic seeds. So their densities and their sizes and their shapes are approximately, as best as we could experimentally, replicate what is happening for, or what we've seen for real eelgrass seeds. I would say the reason that we aren't using real eelgrass seeds is because collecting them would be really difficult in this facility. And so we're actually able to collect these magnetically so that we can make sure that we recover everything. So as we do repeat experiments, we don't see eelgrass seeds that we missed collecting the first time or count them twice. So that's why we're not using the real seeds. All right, and we are going to show results from 3 different canopy geometries. So we have a canopy geometry where we have released the seeds inside of the canopy. So that's about 7 meters down from where this eelgrass vegetation starts, which means that we have fully developed canopy flow. So our flow dynamics we expect to be relatively stationary. They're not transient in time or in space as much in this location. We also released eelgrass seeds at the beginning of the canopy to see what happens if we're really looking at different patches. If an eelgrass seed is released from one patch and then moves over some barren ground to another patch, what happens there? And lastly, we looked at seeds at the end of the canopy flow. So if you're releasing at the end of the canopy and then they're moving over some ground, what does that do? We checked and okay, this one does work. So this isn't exactly the release mechanism that we use for the data that I'll show you, but it is kind of what the seeds look like as they're falling. So these are some of the green seeds that we have, and you can see that it's pretty easy to identify them, which means that our cameras can also identify them, and we're able to track where they go throughout the experiments. All right, so these are some data that we'll put together. In brief, we have the 3 different canopy types. So the first plot on the left is going to show the leading or the full canopy. The one in the middle is going to show the leading edge canopy, and the one on the right is going to show the trailing edge canopy. And the flow speed increases as you go down. So we have 4 centimeters per second to 12 centimeters per second. And so basically we're looking at the distribution of seeds. So if our flow speed is low, we expect the seeds to kind of settle closer. So we expect them to be clustered closer to where they're released. And if the flow speed is high, we expect them to move farther downstream. And I want to point out that this kind of bimodal stuff that we see here is mostly due to not being able to see the seeds because they're hidden between the eelgrass stems. I was really excited that we could have some weird dynamics with like a bimodal distribution. And then I was told that actually it's just camera visibility. So we're still working on kind of filling in some of those patches. And then if we overlay these, we can take a look at some of the differences in the dynamics. So when we overlay these, we see that this blue and this red, which is our full and our trailing edge, don't have as much of a change in their kind of distribution as the green. So the green is the leading edge, and so we see that the dynamics of where those seeds settle really spread out when they're released kind of in this flow that is not as developed as what the hydrodynamics would be if you had released them farther into the eelgrass canopy. So we have not yet kind of looked at all of these parameters that all the parameters that we showed before to calculate all of those non-dimensional numbers for setting up the experiments. We got this data a week and a half ago, I think, so it's still pretty new, and we're working on, first filling in this kind of bimodal part, and then going back to look at all of those different forcings to say, like, when we have this type of flow over a specific canopy, what does that do, and how can we use that to help Trevor determine when and where he should release those seeds? So if we want to know specifically where these seeds might land, then knowing kind of what that flow speed should be, where we should release them, when we should release them, will help us be able to determine kind of where they will go given a certain release point. Right, so this over here is why we have some of those gaps. So this eelgrass stem is what is kind of hiding some of those seeds. So we're working on trying to kind of do this tracking and predicting where the seed would land instead of just taking where their landing location is. And it's proved more difficult than we originally expected, so stay tuned. But we have this bimodal shape due to the eelgrass stem blockage. But the full and trailing edge releases had the most similar results compared to the leading edge, which we saw a big difference in where those seeds landed. Our next steps are to vary the canopy density. So what I showed was just for a single density. And we also are going to work on our seed tracking to both observe kind of where these seeds go when they're hidden behind things, and also to look at kind of look in more detail about how this different fluid forcing influences the seeds movement. All right, thank you.
All right, thank you both for those very interesting talks. We have about 10 minutes for questions. Anybody have a question for either Theresa or Trevor or the two of them together? Saw Will's hand first. Chris is coming to you.
I have a question for Trevor, about So you mentioned that in 2023, it was a horrible year for the eelgrass because it was super wet and rainy. And you said that 2025 was bad as well, but it was 2025, like this summer was kind of the opposite, like very warm and sunny. So I was wondering well, first, if you think the the eelgrass did poorly in 2023 more due to the rain or more due to just like more cloud cover and less sunlight reaching the eelgrass, and then why you think it would have done poorly in 2025 as well.
So go to 2025, I think we did have quite a bit of rain as well through May and into June. And it's really sort of peak, I mean, that's sort of when you're expecting your beds to be coming up and establishing. And because of that, we were going out trying to look for seedlings in early May, which we were anticipating seeing, and there was just sort of nothing coming up through the mud, and that continued on well into June. So I would say that while 2023 was like record-breaking rain through a big point of the growing season, 2025 also had a lot of rain during a very critical developmental point in the eelgrass. But just going back to the question specifically to 23, I would say it was the rain. We would be going out there and there was absolutely zero visibility in the water. We were only standing in like maybe a foot and a half of water when we were planting. So our site over the Lamprey looked like a marble countertop. I mean, there was absolutely zero visibility. So I would say it would be definitely precipitation.
Thank you.
All right, many hands up, Brent.
Yeah, Trevor, I was just curious if you could, since you kind of ended on seeds and you're doing pilot, if you could just elaborate a little bit more on the differences in labor between some of these methods. I was just really curious, and I'm sure some others out here might be curious too, of doing these turfs and collecting adult shoots versus collecting seeds and putting them out. Like what are some of the devil in the details for those?
Yeah, thanks for that question, Chris. I mean, Grant, next year, maybe Chris will give you like 30 minutes to go through it, because I'd love to dig into that kind of detail. The amount of effort that goes into it is actually quite a bit less. When adult transplanting, there's a tremendous amount of work that is involved in both the harvest and the planting themselves. It's a lot of in water time. You're really tied to these low tides unless you're using scuba, which we were really trying not to. It's a tremendous amount of work and money, and very, very difficult to scale up into. On the other hand, seeds, from what we've seen so far, even though, I mean, we're still sort of in this piloty phase, the amount of effort that actually goes in is a lot less. The reproductive shoots themselves are easy to spot and easy to pick in the meadow. These things want to senesce, it's easy to harvest. We go out there, it's a very pleasant days harvesting on snorkel. We bring all of it back and we just sort of spin these plants down through flow through tanks for a number of months. And then we process out the seeds on dry land. And I haven't seen any, there's been a lot of different systems that are working with seeds. I haven't seen any great results that really beat just sort of going out in the boat and throwing the seeds out into the water at this point. So even planting them is a lot easier when it comes to effort.
More questions? I thought a hand over here.
Thank you both. That was a very interesting talk. I have a question for Terry, which is that is there kind of like an ideal travel distance that you want to see in your lab? Like, is there a trade-off between it traveling out of a stabilized sediment zone? I guess my assumption was you want it to travel far, but is that something that you're considering? Or I guess, is that something that you see?
I don't have a distance that I'm expecting it to go, but if it goes too far, then I can't see it with cameras because I only have about 60 centimeters maybe that are actually visible until you kind of lose it with the bit of tank that's reinforced on the outside. I do really, a dream would be to put a camera on the rails that are on the top of the tank. So inside the tank along the top, there are some rails there. So if we could put a camera along there and match it to the flow speed, we'd be able to track it farther. The other issue is that since we do put sediment over the floor of the tank when we put the eelgrass stems in, if your flow speed is too high, you're gonna wash away all that sediment, then you just have a bare panel again. And then you have to kind of shovel, dig out, sieve the sediment. So distance wise for me is only experimental limitations. I don't know how far you want me to go.
So the seeds themselves out in like nature, they actually fall out of the water column very quickly. They look like small stones really falling out. So they don't travel very far on their own. So, I mean, when you talk about the apple not falling far from the tree, it's sort of, but like grass too, so you don't expect it to go far.
Great, I saw hands on
For
this side. of the-
scale, that farthest seed is about 7 centimeters from where it starts on that picture.
Thank you. Yes. Hey, this is Janelle. This might be a question for kind of both of you together, but Trevor had some pictures in his presentation when the eelgrass, or when the seeds grow in the eelgrass, they grow in these spades, like a little packet together. And when I'm out in the field, I'll see those spades floating on the surface of the water, sometimes still containing seeds. So Theresa, I'm curious if you thought about doing any modeling around that and looking at the transport of almost these seed packets instead of the individual seeds.
That's a good question. So I have not thought about doing the transport of the seed packets. If they were floating, that would be very different transport problem with like wind movements, as well as just hydrodynamic forces. Have I thought, I'm not sure how I would scale that to the size that we have in the lab either. Our eelgrass mimics do float though, and we have lost several of them to becoming unattached from their stems. So I have witnessed that, but I haven't thought about looking at that in the lab yet.
Just tack onto that, in terms of Great Bay, I think hydrodynamics play a huge part of that, because I think there's a great point in that these reproductives, these shoots do dehiss and float around. Many of you are probably aware of Tom Lipton's work in the modeling of hydrodynamics in Great Bay. And I think that really offers a great first step and start thinking about where these particles are moving to when it comes to both rack, different flows of water and all the perfect seeds and all of that goes with it.
All right, we have time for maybe one or two more questions. Corey's got the mic.
I have the mic.
Yep.
So I think this is just a follow-up question on the tie between you two talks. Are we looking to, like, is it better if the seed lands in a meadow? Or at the leading edge, or at, is there a place where you want it to land, or it doesn't matter?
The place where I wanted to land, I don't know anything about the growing of them, so I don't know growing wise, like biology wise, what they would want to do. I have some thoughts on this.
Introduce yourself, he was.
Hi, I'm Cynthia Hayes. I'm collaborative with these folks. So, back to your original question about like, is there an optimal distance for how far this seed should go? What I would suggest is that we know from lots of studies of seagrass that tipping points are super important, right? And we know that restoration efforts with low shoot density often fail, right? Because these are plants that modify their flow environment and they facilitate themselves. And one of the ways they can facilitate themselves is if you have a dense meadow, that's going to retain seeds. Right, so again, I predict that there is going to be some tipping point where at some low shoot density, seeds are not retained, right? And that is bad for that local patch. Might that seed go somewhere else and be successful? Absolutely. But in terms of maintaining the patch, I think we do want the seeds to stay close. That's, I don't know.
Trevor, anything to add?
I had this conversation a little bit earlier, and I'll probably have it in the small group discussion too. I will just say that I completely agree with Cynthia. But in terms of funding, funders like to see new acreage created. I would love to start going out and seeing these beds that we see resilience falling and saying we absolutely need to augment these. But when it comes to the actual practicing of doing this restoration, finding funding for it, I think there's multiple conversations that need to be had with it.
All right, one last question.
Okay, just an easy one. So speaking of tipping points, you've mentioned a couple of correlative years with high rainfall early in the season. Overarching all of that is the consistently increasing sea surface temperatures in the Gulf of Maine. The last 5 years, 4 of the last 5 years have been the hottest in the Gulf of Maine. We see Vibrio populations exploding and things like that, so there's gotta be a thermal tolerance that would at least stress the eelgrass. So is that something you can comment on?
Yes. Yeah. Yes, there absolutely is. Generally, it's at about 25 degrees Celsius, sort of that range where it really starts stressing the plants out. And you start seeing mortality after that. I can tell you that out on the coast, we almost never see the water getting to that hot, but in Great Bay, we do on a regular basis during the summertime. So the stressors in the Great Bay system are high, and the grass that grows out there is really the dandelion of the eelgrass world.
All right, that will be our last question, just to keep our agenda